Good evening. Hi, my name's Ted Goldstien, and I want to welcome you to the inaugural UC Santa Cruz Live in Silicon Valley event. Thank you very much to Stanford University and all the other co-sponsors for hosting us here this evening. Tell you a little bit about myself. I'm an alum from 1983, Oaks and Stevenson College. And how many alums in the audience? Excellent. Nice to see a fair showing. And parents of soon-to-be alums or alums in the past? Great. Thank you. Welcome. My own interest in genomics began at Santa Cruz back in 1980, and I did some work with Harry Noller, who was and still is a great professor of RNA biology, and learned the incredible power of it. But there was no place to study bioinformatics yet. And I had to have a career first in Silicon Valley, where I worked for wonderful companies like Sun Microsystems and Intel and some startup companies that you've never heard of, but only briefly had a flash in the pan. And then finished my career at Apple, where I was Vice President of Development Technologies and helped make Xcode and iOS. And that was great. But there was something very unfulfilling about that brought me back to Santa Cruz to come back to work and get a PhD in biomolecular engineering. And that's probably the same things that interest you about this amazing field, that there is just it has a richness to it, a potential, and a way to touch all of human life and all of the biological world that we are living part of. And tonight we're going to share with you a little bit of that amazing story on this. And so I think if you're interested, as I am interested in this, then you'll probably want to join us for the kinds of wonderful events that we plan. We're forming here in Silicon Valley a new chapter, the Silicon Valley chapter of the Alumni Association. And we're going to sponsor the UC Santa Cruz Live events, both here and elsewhere around the Bay Area and in California, but really focused around UC Santa Cruz, about the researchers at Santa Cruz and researchers here in Silicon Valley, that will have panel discussions that bring the best of what's going on in Santa Cruz. And so it's a way that you don't have to go back and get a PhD to get some of the taste of it, but you're welcome to come back and get a PhD. Our mission is to bring friends together and increase the visibility of what happens at Santa Cruz and increase this. Stanford, of course, is a wonderful thing, and we hope someday to turn this into UC Palo Alto myself. Feel free to, by the way, to tweet about tonight. There's a hashtag to use, hash UCSC Live, and you can use that hashtag to ask questions. We'll be monitoring that as you go. I want to introduce as well David Hansen and Mike Rippey. They are also working we're working to form this chapter. And so at any time during or after the event, please let us know if you're interested in helping us out and being part of this incredible thing we're trying to build. And with that, exits in the rear, as they say in the airline. And let me introduce Lisa Krieger. Lisa is a journalist with the San Jose Mercury News. She is award-winning in science work and environment work. And she writes on a variety of subjects ranging from the White House to the water problems here in California. And she will introduce the rest of the evening. Thank you so much.
Thank you, sir. Thank you. Thank you so much. And thank you to all of you who are readers of science journalism here in the Bay Area. Thank you. I'm privileged to be at a paper that still really values science writing, and I hear from a lot of you as readers. We're also privileged to be in a place with so many amazing institutions, so I hope to learn tonight as well and use some of this in our future coverage. So when I think about sequencing and genomics, the one other technology that comes to mind for me is the Hubble telescope, which I've written about. And the reason why is that both tools are just opening amazing universes, places that were very unknown and very mysterious, but they explain who we are, where we came from, and what our future is. And a lot, like the Hubble and genomics, it's really teaching us things we never thought were imaginable. Probably a lot of you remember, it wasn't that long ago, year 2000, when it was, what was inside a cell was all our dark matter. We really had very little understanding of what lay inside. And medicine back then was very much a sort of find it and fix it, understand things in a very large, gross, undifferentiated way. And that led to some beautiful cures. We do have beautiful cures, but for 2/3 of diseases that are very complex, it's been quite insufficient, as I know you all know in your own private lives. And we really needed more. So that changed in the year 2000. I was privileged to cover this. Francis Collins and then President Clinton, Tony Blair, Craig Venter announced a sequencing of the human genome, and that was just transformative, completely transformative. So suddenly we understood that there's this huge jumble of genes that control a lot of who we are and how we work. But then here's the hard part. I initially, like a lot of people, thought it'd be one gene, one disease. We'd find the genes, we'd find the diseases, problem solved, right? Not the case at all. It's not, it's not even this long, linear, beautiful pathway of genes that lead to things. What we're discovering, which we'll hear tonight, is it's many pathways, many relationships, all relating to each other, and very, very complicated process. So this is when I always feel like I need to speak on behalf of Mother Nature and defend it a little bit. We're complicated for a reason, right? If we were hardwired, we wouldn't be able to respond as beautifully as we do to changes in physiology, changes in the environment. And there is something really gorgeous about the complexity. But because biology is so complicated, that means disease is complicated. And thank God for math, right? Yeah, thank you. And computers. So what they're showing us is not only what are these sequences and how do they relate to each other, but how do things go wrong when their relationships change. So what we're going to hear about tonight are really some leaders in the field. I can't tell you how privileged we are to have these folks here. When I saw the lineup, I knew we'd be in great hands. They're going to talk about big data, how we're translating this big data to learn about the underlying mechanisms and how things go wrong. So I want to just tell you a little bit about the format of tonight. We'll have each speaker give several minutes summary of what their research is and what their findings are. And then we're going to go to a Q&A. So I'll ask them some questions, maybe 20 minutes or so. And then the floor is yours. So I encourage you to be thinking of questions while you're listening to them speak. And then and we've got a microphone over there that you can go to address your questions. So we're going to start with David Haussler and Olena Morozova, who are at UCSC. As you know, UCSC is not a medical institution, right? There's no hospital, there are no clinics. We don't think of it as a medical place, but and this is huge, it's a computational place. It's an analytic place. And what they're discovering there is utterly changing the practice of medicine. So with that, I'll turn it over to David and Olena.
Thank you so much, Lisa, for these wonderful remarks. And I love your big picture, because we think of genomics now as a telescope into a new world that we couldn't see before. And when we first approached this in the Human Genome Project, we had no idea what we were actually going to find. And it was a privilege for Santa Cruz in particular to be part of this. We weren't chosen a priori because we're a big med school. We contributed the informatics. In fact, in the year 2000, when we assembled our first draft of the human genome, as you mentioned, I always like to tell the story for any Santa Cruz audience. It turned out that there were because of the race between the public project, which Santa Cruz was a part of, and the private project that was racing us, the company Solera, there was a hasty decision to try to put together this first draft as quickly as possible. The public project wasn't prepared for the computational challenge, and it was a graduate student named Jim Kent that actually put the first 400,000 pieces of DNA together to give us our first draft of our recipe. So this was an amazing event that happened at Santa Cruz because of our computational expertise at that point. And that was really the birth of the UC Santa Cruz Genomics Institute as a seedling that has now grown into an official institute. And for me, it's a profound thing. And again, from the big picture, it's a 3.7 billion year struggle. The first idea of life in the ancient times was a great idea, and to use DNA to replicate and carry on the message from parent to offspring was a great idea. And the result of all the triumphs and stumbles of all of our ancestors over so many eons is a 3 billion letter text that we all carry inside every cell of our body. And we went through a profound transition at the moment in which we, as a species, read our own DNA. Enough information to carry a single cell all the way into an adult body and mind. So this is an extraordinary opportunity now that we can actually read this message and use it to understand life, the processes of life, and to understand our own health and our own being at a more profound level. The technology that enables this is absolutely incredible. And so I think we have to underline this as a technological revolution that allowed the science. And I'm going to pay tribute to my colleague, Ewan Ashley, who remarked on calculating the improvement in genomics. He looked at the figures. The first human genome, the draft that we talked about, cost hundreds of millions of dollars. And today, you can get your genome sequenced for about $1,000. And so his analogy, to get you to understand how massive, disruptive, and incredible that change is, he said, look, if the automobile industry had approved at that rate, a Ferrari would cost 40 cents today, right? So this is really important to think about how incredibly new and what amazing potential we have in reading this message that was carried to us through all of those struggles of our ancestors. We want to use that message to better human health, and it has enormous potential to do so. Santa Cruz, in light of actually not being a medical school and not being competing with the other major medical schools, has been chosen now multiple times to be the data center for big data. We were first chosen to be the center for the National Cancer Institute's data, and we built the largest database of genomes in the world for the purposes of research in cancer. That project was one of many projects that put us on the map in terms of biomedical research. The UC Genome Browser, which has information about human genomes and human variation, is visited by hundreds of thousands of researchers every year. We get 1 million page hits a day at this browser. So all of these things that we've created, the informatics tools that let us understand this heritage, have been instrumental in taking us to this new level, taking us through this portal to a new world where we can read our own DNA and interpret it. What we want to do here is to use that for health. And so we'll be talking next about cancer. And I have a video that's very compelling that will be my introduction to my colleague, Olena Morozova, who will talk about the actual programs that are happening right now to use the genome to help with cancer. So let's run the video, and then I'll turn it over to Olena.
My name is Libby Kranz, and I'm the founder and executive director of Unravel Pediatric Cancer. Our goal is simple: to unravel all cancers. We believe that you start with the hardest to understand, the hardest to treat, and the ones most dramatically underfunded, pediatric cancer. I was thrust into this life when the eldest of my 4 children was diagnosed with DIPG, an always terminal brain tumor, on her 6th birthday. Even with treatment, she passed away 3.5 months later. It was never a fair fight. The doctors treating her had no arsenal of weapons to give us to protect our daughter. It is because of that I am, and our foundation is hopeful to partner with CKCC to raise awareness, funds, and to help engage parents. Pediatric cancer is an ideal fit for the California Personalized Medicine Initiative because every patient is so unique and every cohort is small, but has profound effects on the entire cancer community. Chemotherapy was begun by Dr. Farber, a pediatric oncologist, and a current example is Termoplant, created by a pediatric oncologist and now being used in adults. The need is great. We are obviously the worst case scenario, but unfortunately with pediatric cancer, that's not the end of the story. It's not black or white. It's not that kids just live or die. Survival is a never-ending battle for the kids, with 3 out of 5 survivors facing long-term side effects. Better than dead is not good enough for our children. To be able to treat each child, not as a type of cancer, but as an individual, can change both of those outcomes. In turn, saving entire families. To be able to connect each child to the correct kind of treatment would, well, it would be a complete game changer. I urge you, and I ask you personally, on behalf of my Jennifer and the 7 kids that will die today from cancer, to help us.
So, as you heard, children do get cancer, and unfortunately, despite the statistics that we hear, that 80% of kids that get cancer are cured, despite that, this is definitely not a solved problem. So you've heard of diffuse intrinsic pontine glioma, or DIPG, which is the disease that Libby's daughter had. So that disease has 0% survivors today, 0%. Nobody, I don't know, and if you Google DIPG, there is no survivors at all. And there hasn't been any progress in the last 50 years. Radiation is the only thing we can do for these kids. In addition to DIPG, another problem is children whose cancer recurs after standard treatment. So the way that we treat children in the clinic today, and Alejandro, who is here, can tell you more about that, is basically we use really brute force approaches. We throw really toxic chemotherapies, radiation, bone marrow transplants at these children, and we hope that we kill the tumor before we kill the child. And so once the tumor comes back after that, those tumors usually don't respond to anything else. So cancer is actually #1 cause of death by disease in our children, and it kills more children than every other disease combined. So on a good note, we're doing something about that. We have a very exciting project called California Kids Cancer Comparison, which is one of the 2 official demonstration projects of the California Initiative to Advance Precision Medicine. And so the goal of this project is to learn how we can use genomics to provide new hope for difficult-to-treat cancer patients, childhood cancer patients. That includes patients that have DIPG, diffuse intrinsic pontine glioma, and that includes patients whose tumor came back after standard treatment, so relapsed and refractory patients. So we're partnering with 3 clinical trials, and one of them run by Dr. Sweet-Cordero at Stanford. The other 2 are at UCSF and Children's Hospital of Orange County. And so all of those trials are using genomic sequencing, genomic analysis to understand the tumors of these patients and to see what we can do to suggest new treatments for them. But it turned out that genomic sequencing is actually just the beginning. So generating sequencing data now became affordable. We have great advances in technology that allowed us to generate sequencing data, and it now costs less than $5,000 per patient. But it's just the beginning. And just to illustrate how that's just the beginning, I would like to share one anecdote with you. So this is a patient, our first patient, or CKCC patient 1 that we had. And this was actually a partnership with our collaborators in British Columbia and Canada. So that patient is an 8-year-old boy, and he had a rare sarcoma tumor. And he was enrolled on a trial that was going on in Canada where the genome of this tumor was sequenced. And so it was found that there was a particular mutation that could explain the cancer. But the problem was that there was no drug right now to treat that mutation or to kill that cancer. And while it's interesting from a biology point of view why that tumor had a mutation, for that particular patient it's back to square one. Even though we have the genomic sequence, we can't do anything about it. And so our collaborators in Canada shared the genomic data with us. And what we have done at UC Santa Cruz is we were able to compare that patient's genome with other genomes we had in our database, which included tens of thousands genomes from adult and from pediatric patients. And then when we did that comparison, we actually saw that despite the fact that this was a sarcoma, this patient's tumor was similar to another tumor type that had a treatment associated with it. And so we could identify this drug that we then communicated this analysis to the oncologist, and the oncologist felt that it was a good idea to try this drug. And so the patient responded to the drug. Now, not to oversell this, unfortunately, the tumor eventually did come back in this child, illustrating that this is a new approach. We need to do more, and this is just the beginning. But this really illustrates how this is a paradigm shift in medicine. Now, computer geeks like us at UC Santa Cruz, we can partner with oncologists and we can actually produce something useful that clinicians can use today in the clinic. And so we're very excited about that. And we're very hopeful we can use this approach to help 150 patients in California who have difficult to treat tumors. And of course, our big dream is to do this for all of 500 children in California who failed treatment today, and then eventually for every child. Thank you.
Thank you. Beautiful, thank you. We have some really nice bios of our presenters tonight, so I will not go into detail. But our next speaker will be Dr. Alejandro Sweet-Cordero, who can tell us about the patients in the clinic that he's seeing.
Thank you. Thank you for being here tonight. I'm very happy to be here. I am a UC alumnus. I graduated from medical school, UCSF, and I'm probably a honorary alumnus of UC Santa Cruz since I grew up at Merrill. When I was a child, my dad is a professor of history at UC Santa Cruz, so I lived at Merrill College when I was a kid, so I think that qualifies me as a UC Santa Cruz alumnus. So I thought I'd start by just telling you a little bit about how I got into this. I did my pediatric training also at UCSF and then went to Boston to do an oncology fellowship and did my research part of my fellowship at MIT, where I really focused most of my work on studying cancer in mice and doing cancer genetics in mice and started doing genomic analysis using some of the tools that you've been hearing about back before sequencing when we were doing what are called microarrays, which is sort of an earlier version of gene sequencing. And when I came to Stanford, I continued that work, and that work has gone very well, and we've continued to do that. But I'm also a pediatric oncologist, so I also treat patients at Packard Children's Hospital. But I always felt like there was a little bit of a divide between what I was doing in the lab and what I was doing in the hospital, and they weren't really very well connected. And one day I was taking care of a young woman who had relapse and who was having a difficult time with her treatment. And I was really frustrated because I realized, here I am writing the chemotherapy orders for this patient, giving the same chemo that we used to give 30 years ago, when I know that there's been this huge revolution in our ability to analyze genomes, as David described, but we're not really using that to help these patients sort of in real time. So I really decided that I needed to change the way that I was doing things, and I started getting involved in this effort to apply genomics in real time for our patients at Packard Children's here, just a few blocks away. So that's the work that we're getting engaged in now. Fortunately, we've been able to get the very strong support of the leadership at Packard Children's School of Medicine to really develop this effort to try to sequence all patients who relapse at Packard Children's or patients who have high-risk disease where we know that our chances of having a long-term effect on the patient's life are small, and really try to change that with the power of genomics. And we were going along doing that kind of on our own with our own resources until about a year and a half ago when I met Olena at a meeting, and then through Olena met David. And it's really been fabulous and fantastic. It's really elevated us to a whole nother level. And I'm really, really excited to be here tonight and to be a part of this collaborative effort. Certainly Stanford has a lot of resources, but as David described and as Olena described, I think UC Santa Cruz just has an unbelievable repository of information and a repository of knowledge, and not just a repository, but the ability to analyze that data. So I think together with the patients that we have here at Packard and the infrastructure that we're putting together to actually in real time being able to extract the tumors from the patients, do the analysis of the making these sequencing libraries so that we can then generate the data, and then working closely with the group at UC Santa Cruz to analyze these data sets, I think is really, really exciting. One of the things I think is really powerful is that if you think about pediatrics, I don't know, you hear about personalized medicine, people talk about how we need to put people in so-called buckets based on their genomics instead of based on what the tumor is like. Well, think about pediatric oncology as like not a bucket, but a little thimble. So every patient is almost like, their own disease, right? I could give you examples of how, all the patients that we take care of are each unique in their own way, and they're suffering in a very unique way. And you heard a very compelling story tonight, and we could give you many more. So our ability to really understand these little small, tiny little buckets is magnified just exponentially by the collaboration that we have with UC Santa Cruz, and we're very excited to move that forward. And I think that over the next 6 months, 12 months, year, 2 years, we're going to make a very profound impact on the care of these children. So thank you for being here tonight.
Thank you. Next, we'll hear from Gini Deshpande, who's CEO and founder of the company NuMedii. She's an expert in taking these technologies and turning them into products and services.
Thanks, Lisa, and thanks everyone for the opportunity to present a little bit about NuMedii to you today. So as Lisa mentioned during her introduction, we are shifting away in the pharmaceutical world from this one target, one drug concept, and as Lisa mentioned, we are not looking at these now as single pathways, but really pathways of pathways. And I think big data is really enabling us to take a very different view or very different look at disease biology and really starting to understand that. And particularly in areas like cancer, I think the notion that we will be able to come up with one treatment and then the patient's going to either be cured or their disease is going to halt progression, I think is a notion that we have to get away from, and we are starting to get away from it, because I think as patients' diseases progress, their profiles change and the treatment paradigms will need to change. So obviously all these wonderful scientists and clinicians here are helping to make these changes happen. Hopefully we'll see that adoption happen in the healthcare space. What NuMedii is really focused on is enabling drugs to reach these patients, because I think one of the things, as Olena pointed out, there's a lot of information we're collecting on patients. We're identifying a lot of mutations. There's about 50 mutations for which there are targeted therapies available today. And from a drug development perspective, if one were to look at trying to develop a drug for every single mutation that we identify, that's going to be a really, really difficult challenge for the pharmaceutical industry. So we started NuMedii with the view of leveraging all the information that we can about both diseases and drugs and helping develop drugs in a much more expedited fashion for patients. And we thought, why not start with drugs that already exist? So those of you who might not be familiar with drug discovery and development approaches and historical failure rates, it takes about 10 to 15 years and about $1 billion for one drug to make it out the door. Now imagine, trying to do this for, as Alejandro pointed out, for, one thimble when you're trying to develop something for a bucket. It's just not practical. So what if we can leverage all the existing drugs that we have and find new uses for them through big data? And that's essentially what NuMedii does. And we look at sort of patients as a spectrum. You can look at the buckets that Alejandro referred to, or the thimble, and the approaches that we take by connecting drugs and diseases essentially can be applied to both categories. So we find new uses for existing drugs for large disease indications like ulcerative colitis and psoriasis, which are two of the programs that NuMedii is developing, or for individual patients. And one of the reasons we're really excited to partner with the California Kids Cancer Comparison is because there may be a potential for us to identify existing drugs that could potentially benefit patients that may be outside the sort of traditional realm that we're thinking of, where they may be targeting a specific mutation that we know, and looking at off-target effects of drugs using a big data approach and helping benefit these patients. So we're very excited to collaborate with them and hoping to further progress this collaboration. So thank you very much.
Thank you. Next, we'll hear from Dr. Atul Butte, who's a real leader in this field. You've probably heard his name. He's been quoted all over. He's involved with NuMedii, and he's also director of the Institute for Computational Health Sciences up the road at UCSF.
She calls me a real leader compared to these fake leaders here, right? No, it's a real honor and pleasure to be here. It's great to be on the Stanford campus again. For those that know, I was here at Stanford for 10 years, been at UCSF for 10 months. They're probably towing my car as we speak. I had to park next to the curb there. I think you parked there too. My car will be gone. But I think they still love me here. I don't know. They still at least tolerate me here. But it's a real pleasure to be here. So you've seen big data. You've heard about some of this big data. And you've also heard this term precision medicine, right? And it's a kind of odd term, precision medicine. You know, it makes the doctors really nervous, precision medicine. We're heading to this era of precision medicine. Because that means, by definition, the last thing we were doing was imprecise medicine, right? So it kind of makes us nervous, right? So what is imprecise medicine, right? What were we doing that we wouldn't call precision? Well, one way to think about imprecise medicine is just how do we characterize diseases, right? What is our list of diseases, right? Do we have, is every disease in a list? Actually, we make these lists. Any of you that have gone to a doctor back in the paper-based days, remember that big grid on the back of the sheet you're supposed to hand back to the receptionist to check off these codes? Those are international disease codes, ICD-9 codes. That's a list of diseases. And in some ways, that's one way to just think about how precise our medicine can be. So I carry around literally in my bag, books of these kinds of codes. I love these old books, right? People throw them out. And ICD is just a great example of how bad medicine can be sometimes. I'm a physician, very much part-time now. But it's an example of how bad things go. I mean, this is ICD-2. Forget about ICD-9 codes. This is 100 years ago. This is second revision, Paris, 1909. I carry this around to just remind us how imprecise medicine can be. And it's amazing. 100 years ago, and you can actually find these books on Google Books, this book is kind of written the same way our current ones are. Use code 76 for this ear disease. Use code 77 for pericarditis. That's inflammation of the heart. And then literally, this book says, here on page 141, it says, well, if you run into a patient who died because of a visitation of God, use code 189. I mean, it says it right here, right? This is imprecise medicine, okay? And this is 100 years ago, and we can easily laugh at this, but we should actually laugh at what we have now, okay? So big thing that happened last year is we moved to ICD-10, okay, the 10th edition. And, actually we in medicine made a big fuss about this because we make fun of all these precision codes. There's a code for being hit by a whale while surfing, right? I mean, maybe that happens in Santa Cruz. It doesn't happen here. You're going to need that code there, right? You might need that code, right? And it's a public health menace, right, over there, right? Not here. But ICD-9 is pretty bad. I mean, so when we laugh at ICD-10 and these codes, ICD-9 is what we should laugh at. I mean, when you think about lung cancer, right? Do they have molecular coding for lung cancer? Do they even use the microscope? Is it small cell, large cell, adenocarcinoma? No. Do you know how they code lung cancer? Left lung or right lung, okay? Not kidding. That's the code, right? ICD-10 isn't even that much better. And what we need to do is go to the modern molecular revolution for diseases. I got my start in this career about 15 years ago, analyzing these kinds of chips, gene chips. So I thought I'd bring some props here, right? And so we can use these kinds of chips. We have fancier machines that look like glowing lights and Cylons and stuff, but this is one I can carry in my bag. And when I got started about 15 years ago, we could put a sample of cancer on this and just get a readout of the genome, right? It's kind of amazing, and they're a couple $100 each now. This one's from a company called Affymetrix, which is just down the road in Santa Clara. Sadly, the company just got bought a couple weeks ago, so this company doesn't even exist. But now, when you get tired of measuring them one by one, we measure them in the 96 well plate, and that's this one. In fact, this one is so cheap I can carry this in my bag now. And then when we got tired of measuring them 96 at a time, we measure them in the 384 well plate, right? And look how tiny that is. And I cannot more clearly illustrate exponential growth in this data than to show you these plates, right? What's the next one gonna look like? I don't even know. They're actually not even in plates anymore. We got these machines to do this, right? And this is the modern revolution. We're going from old books like this to this, right? That is precision medicine. Now, we've heard about precision medicine because President Obama even mentions precision medicine about a year ago in the State of the Union address. It's kind of amazing a president actually talks about this stuff. They talk about cancer moonshots now. Let's make sure kids benefit from that too, right? We got to make sure of that. But actually, I'm really proud that I get to work with the state government actually on precision medicine. So President Obama mentioned this in 2015, but Governor Brown mentioned precision medicine in the State of the State address a whole year earlier in 2014, in the State of the State address. Now, I will be the first to admit, I didn't even know Governor Brown gives a State of the State address. I kind of, now I know to look for this, right? But it's there in 2014, at the end of the State of the State, he talks about precision medicine. And what he did is he created $3 million, which even in Sacramento, they call this budget dust, okay? $3 million, at least get this initiative launched. I'm proud that I get to help launch this. And they gave it to us in a hurry, so we had 90 days to try to figure out teams to approach. I think we were bombarded with 50 different applications from all over the state. I'm so happy these guys got one of these, okay? And it's just $1 million, $1.2 million. It's not a lot of money, but it's enough to at least say that we can do something. Now, Governor Brown's proposed $10 million, which is a little bit more budget dust, I guess, compared to the billions. But it's at least showing that we are in an amazing state, and we can do more about it ourselves too. Not just the big picture, not just the whole country, but our state should do something about this too. Maybe it's as important as stem cells and some of the other things we can invest in here. So this amazing time to be in science. You can see a lot of science is digital now. It's not, dissecting frogs with formaldehyde. It's actually writing a little bit of code now. And so if you ever even think you're too old to get into this field, you're probably wrong. But if you do think that way, get your kids in this field, okay? Because it's the most amazing time to be in science working on important projects like this. So thanks again for coming out tonight.
You have a daughter, don't you? Thank you, Atul, who has a daughter who's interested in science. You can ask him about later. Our next speaker is Matthew Bookman, who graduated from Stanford, studied computational biology and computer science. He's now at Google, and he calls himself a solution architect, which he'll tell us about.
Thank you. So I'm a solution architect on the Google Genomics team. In 2010, I left my position at Cisco, where I was a software engineer, my son had just been born, and I wanted to commit to being more of a full-time father. And I also had this dream that maybe I would be able to do something that was more, that was going to tackle one of humanity's great challenges, save the world with software. And so it turns out that there was a project, oh, my. Sure.
Luckily, it's behind the podium. So
Yeah. Okay. Can I keep talking? Okay. So it turned out that there was a specialization within the computer science department called computational biology, and it also turned out that I had this long abandoned master in computer science that I had left in the 90s, and it was a great opportunity to go back, and so I did. And so it was a fantastic program, truly life-changing. In this in this program. We read papers from David Haussler. We used tools, as he's mentioned, from the Haussler lab. I read papers from the Butte lab and took classes from Atul. And I can say with all sincerity, thank you to both of you for your part in making possible what I'm getting to do today. So after graduating, I interviewed with several organizations who were doing genomic research, and all of them said the same thing: we have to move to cloud. The data that we're working with is too big. We just finished doubling the size of our compute cluster, and we need to do it again. And the data is big, it's difficult to handle, it's difficult to share, it's computationally expensive. Our scientists spend time doing a lot of IT and not doing science. So what did they mean by moving to cloud? So Google, I can speak to Google Cloud, has spent the last 18 years building out a hardware and software infrastructure to deliver services worldwide, handling the largest data sets in much of the public sphere. They process the data and deliver results to people around the world at incredible speed. And so Google Cloud is taking that infrastructure, both software and hardware, and making it available to the public. You have the opportunity to use most of the same tools that Google engineers use. So how do we apply this to genomics? So a good example is a project that I've been working on with Autism Speaks. They are looking to build a genomic database of 10,000 whole genome samples. They get their sequencing done by a company in Korea, Macrogen, who pushes data directly, after doing the sequencing, pushes the data directly into Google Cloud. And as soon as they're done, the data is available to the researchers in Toronto at the Center for Applied Genomics. No shipping of hard drives, no saturating anybody's network link. It's fantastic, and everything moves much more quickly. I'll give one more example, which is the data is computationally intensive to process. It's common for processing a whole genome in 2 to 3 days, a single whole genome. And so how long does it take you if you've got 500 samples? Do you do them sequentially? Does it take you 1,000 days, which would be about 3 years? Maybe you have a decent-sized compute cluster and can do 20 at a time, and you'll get done in 3 months. Well, with cloud technologies and with compute cycles from Google, if it takes you 2 days to do 1 sample, it takes you 2 days to do 500 samples. And this is what we really want to do, is to take this infrastructure, apply it to science and health and medical delivery, and dramatically shorten the amount of time that is spent waiting arbitrarily for computation, and really help scientists do science. Thank you.
Thank you. Fascinating. So now we're going to arrange some chairs up front and get everyone together, and I'll ask a few questions. So, just for folks who may not be familiar with how this works, talk about sequencing and how do you find a meaningful pattern in all of this data? We all, lay people read about sequence this, sequence that. How do you look at that and then identify? Is there something here? Does a small change mean a big problem? And
Yeah, this is the fundamental question. What the hell do you do with 3 billion ACs, Ts, and Gs? So first of all, we're all of us 99.9% identical. So we are incredibly similar. And you can compare our genomes to the genomes of other species, and we also find remarkable similarities. And this is not surprising. It's because we all share a genomic heritage with our ancestors. So the number one way you understand a genome is to compare it to other genomes. So by doing that genome comparison at scale with the kind of resources you heard about from Google, we are able to run these massive comparisons now in the cloud, more than we could ever do before. And we start to see now, sifting through these billions of bases and thousands of cases now, patterns. And it's something that no human can do by themselves. It's only machine-aided intelligence that will actually understand the patterns that are in our genome and connect that to disease. So it is the day of the geeks. It's a time for computer scientists to step up and help medicine. Because it's not enough to do it the old-fashioned way, and I think all of the presenters made that point in a brilliant way.
So is there a standard genome that you compare it to, or how do you know what specifically is wrong
Right.
and why it's broken?
That's a good question. So when we first posted that genome on July 7th, 2000, that was the standard genome. At that moment, that became our standard human genome that other genomes would be compared to. Now, we have improved that standard steadily in the 16 years since that day when the first genome was posted. Now, we're on a version of the genome that has much more completeness. There are still strange places in the genome that we haven't been able to penetrate, but we're at an enormous fraction of the genome. And right now, we are at Santa Cruz, interestingly, in another project to get away from the idea that there's one standard genome. So what's happened in the last 5 years is that we've discovered that by choosing an arbitrary individual to represent our standard genome, we were biasing all of our analysis. So if you are from an ethnic population of humanity, and you're looking at this standard genome, and that standard reference is from an individual that's not of the same ethnic population, your raw DNA reads don't map as accurately to that standard. It's a tiny, tiny effect, but over billions of bases, this can be important. So actually, we are now creating at Santa Cruz, with funding from the Simons Foundation and also the Keck Foundation, a new standard genome, which will have all of the different human ethnicities and their human variations expressed in one standard. And I won't go into the mathematical details. It's called a graph genome, and it's got some bells and whistles to it. If you're a computer geek, you'd love to hear about it, but I won't bore you with it tonight. But it is going to be a very, very important thing, and it will allow the precision medicine that Atul was talking about. It will allow us to name the human variants rigorously in a way that won't change as we improve the genome. It will allow us to use those names to communicate between physicians and scientists that this patient has this specific genetic change at this specific point that has a name that we are not going to confuse, and it's equally readable as any other change from any other ethnicity on the planet.
Might the day come when we could link that to medical records?
That would be a wonderful day.
Anonymous, of course, is good. So that
Yeah,
everybody is-
I think Atul should
Yeah,
address
that's an
that
important
one.
point.
Yes.
I mean, what really kind of makes this field really impressive and really interesting is you have two kind of multi-billion-dollar efforts colliding here in this space. So one is obviously this whole genome revolution. I mean, the first genomes really took $3 billion, and we're spending millions of dollars and thousands on each one, but you have $1 billion effort in biotech and genome sequencing. And then you got another multi-billion-dollar effort on electronic health records, right? That means getting rid of those paper sheets and getting us all coding what we're doing, coding what we're measuring, and collecting that on populations and interoperating someday between all of our sometimes competing medical centers. And isn't it amazing when two multi-billion-dollar efforts collide? And that's what this is, right? We've spent a lot of time talking about the molecular side of this. Right. Mm-hmm. And precision medicine is, half of it is learning what's going on with you and your disease and your cancer. But the other half is learning, well, what did we do with everyone else that looked like you before? And that's all captured in the electronic side, on electronic health records. What did we do last year for patients like this? Have we ever seen a case like this before? We can't look in paper records anymore. We've got to do that electronically. That's really, it's this intersection. Makes it amazing.
You could link clinical outcome or responsiveness
That's
to
right.
chemo or whatever based on specific genetics.
That's right. I mean, so in some ways, we can kind of train computers to play chess, right? Because they can see so far ahead. We can now train them to play Go. Even I don't know how to play Go, but I guess the computer knows how to play Go now, as of a couple weeks ago. But you think about, and I don't want to trivialize this, but every case that comes into hospital, it's like we're starting a chess match, and someone somewhere knows how this ended out, right? I mean, and we're not even keeping track of what we're doing here, and then we try it again, and we're not learning, right? And this whole system is what people call learning healthcare system. Like, let's keep, like every case, every patient, we should be learning something from. And sharing these records, of course, keeping people, data private and secure, but let's learn from every single case that comes through all of our medical centers and use that to help us with the next one.
Yes.
Thank you, which leads me to a question for Matthew, and that is, you think of the Alexandria Library that had every book, and clearly you're doing that with scanning with Google.
Right.
Could Google hold a massive library of every genome that's been sequenced, and then we could share it, right? And we could learn. So now it's very competitive, and there's silos of information.
Right. So I certainly would say that technologically they could. Then there's issues of who maintains and controls the data. So far our approach with Google Genomics is that we would support others in science. Whoever owns the data, they own the data. Google does not. But I think that this should be the direction that we go, is to use these resources that are available to do these massive scale databases. Exactly.
I want to jump in here, and first of all, I want to commend your Google Genomics team for all that you've done and the open attitude that you've taken. There's a little bit of a backstory here. Matthew and his team are one of the major participants in a global initiative called the Global Alliance for Genomics and Health. This is the preeminent organization for standards for how to represent and exchange and use DNA data. We are here to revolutionize the medical informatics out of the Stone Age and into the modern digital age. And Google has been wonderful in that. I am one of the 8 co-founders of the Global Alliance for Genomics and Health. My other 7 co-founders are distributed around the world. They are preeminent people, much more so than me. I was kind of the geek on the project. And they together have now collected 385 different organizations from 37 different countries. We have more than 600 individual members, and we have created an enormous repository of open source code and schemas for representing data so that all the computer scientists could agree on a machine-readable exchange. It's easy as, the things that you think about in the internet, the standards, HTTP and so forth. How do the computers know to talk to each other so well? We're doing that for medicine. And we have 120 companies involved in the standards making process. And it's very different from some of the companies that were involved in medical records who didn't want standards and didn't want to share. When we start working with information companies like Google, they want standards, and they are sharing, and in fact, a lot of the horsepower in creating these is donated time from your team, Matthew. So I really commend this. You put your engineers on, and they are creating open source software and creating standards for everybody to use, alongside 119 other companies that are all agreed to work on this. So this is a huge step forward.
Thank you.
Thank you. Related to that, if we could, talk a little, if you could, about security concerns as massive amounts of data are moved from one place to another. And then related to that, patient privacy.
Right. Well,
Anyone want to take that? Oh, sure, I'll take that too.
Yeah, go ahead.
Sure, why not take that, right? So I think obviously we know, most people watch Law and Order, which is kind of gone, and CSI. You know what you can do with DNA sequence. You can kind of identify people. So beyond that, if you actually have the address and location, you can really identify people. So there's a certain level of health data that also it gets harder to share. So the reason why we can't just make things open is obviously we don't want everyone to know about our health data and what those kind of bytes of data actually mean. So there's extra sensitivity here, which makes things harder, but necessarily harder, right? We're not trying to kind of take any shortcuts here. So in some ways, there's new methods in encrypting data, encrypting identifiers so that we can keep track of who this person is, but we don't need to know who this person is. And that way we can link data sets and link them with the molecular samples later and keep this kind of study going. But it's still a hard problem. There's a problem right now called interoperability. For example, even though I was at Stanford and I'm at UCSF, I get my care at Palo Alto Medical Foundation, which is the third competing element on the peninsula here. So, neither of the two actually. And so, do the records communicate? You know the answer to that, they don't. And we can maybe get to them on an app from Epic, and we can get them on a website, and we can do a little bit of downloading and things like that, but we need to get to a state where we all trust each other, at least enough to share this kind of record data, but do it in a secure way that it's actually protected. But it's a hard problem. You know about healthcare break-ins, a lot of big stories in the last year or two. There are probably going to be more of these. And we all have to do our best to do this, but it's still important. It's still important for us to be able to share the data because we have to learn from each other here, right?
And I'd like to, yes, of course. So we have to share data securely. Yes, there are privacy issues, but I would like to raise this point: the risk of not sharing data.
Yeah.
So patient one example that I talked about, we took this child's tumor and we put it in the context of all the other data. And we actually have a tool, Tumor Map, that allows us to do that. It's based on Google Maps API. And so I don't have a slide, but basically what it does is you have different dots that represent different patients. And then you have this one patient that's kind of in the context of different dots. So those dots are patients who have shared their data with research. And it's thanks to those patients that this one child, we were able to spot that similarity and suggest this new drug. So have those people not shared the data, that child would not have this treatment option. So what about the risk of not sharing?
Yeah.
Oh, well said. Thank you.
Yes.
On a related topic, maybe Alejandro can address this. Do you find insurance companies supportive of this? And since this is the way we're going with healthcare, are there issues in getting children sequenced, and who pays for it?
Yeah, well, that's a great point. I mean, it's a complicated question in the sense that because the sequencing that we're doing right now, which is whole genome sequencing, as people discuss, isn't really approved in terms of a clinical indication. It's not something that an insurance company is going to pay for because it's really very much seen as a kind of discovery tool. So insurance companies will sometimes pay for more directed panels, sort of cancer-directed panels, so there's various companies, Personalis, Foundation Medicine, Caris, various other companies that sell these kind of panel kits. And we've certainly worked with those companies, and sometimes insurance companies pay for it. Other times, actually, we've had a good relationship with Foundation Medicine where they've given those tests at cost. So I think that it's something that probably the adult doctors are dealing with more because they're ordering many more of these tests. I think pediatric oncology is a pretty small sliver of the oncology pie. So I think it's sort of falls outside of the radar for most people. But I think it's certainly something that I think we're gonna need to deal with. And the other issue, sort of related issue, is that as I mentioned, whole genome sequencing isn't approved for clinical indications. There's a process called the CLIA process, which is basically the way in which a test gets approved for as a diagnostic, and that's a whole other area that is still kind of not fully resolved. And how do we make these tests kind of approved by the FDA for diagnostic use? And that's something that's very much sort of ongoing. And I think part of the problem is that the technology. I mentioned this to somebody before during the cocktail hour. Was that I think that the technology and the expectations are moving faster than the reality. So a lot of the reality that happens, day to day in the hospital, is sort of way behind our technological ability to sequence these genomes. And and I think another issue that we haven't really touched on, but I think Gini's presentation is very topical. Is it's great to do all this genomics, and it's great to do all this target discovery. But the fact is that, particularly for pediatrics, we're way behind in terms of actually having drugs that we can apply to these discoveries, right? So that's a huge other effort that has to happen kind of on the other end. Like, once we figure out how to do all this, data analysis and how to get the insurance companies to pay for it and how to link it to the electronic medical record, it's not going to do us any good unless we have drugs to give to the patients. And that's, a huge effort that, and then linking the genomics to the drug discovery effort is another issue. Because right now, most of the drug discovery efforts are not done kind of in the genomics mindset. So that's, there's a lot of work that still needs to be done.
Thank you. Question for Gini related to that, and that is the repurposing of drugs. And are pharmaceutical companies receptive to that? Is that considered an off-label use?
So, doctors are certainly allowed to prescribe drugs in an off-label fashion. They're totally allowed to do that. The challenge, of course, for doctors then is getting reimbursed. And that is a whole conversation between providers and payers, and how that gets handled is very variable. From the drug development standpoint, there's certainly interest in repurposing medicines, but for some reason, pharmaceutical companies have sort of shied away from doing traditional repurposing, which is taking a drug and moving it forward for a new indication. Now, there are companies that have done that very well and very successfully, where they've taken a drug and moved it along for multiple indications. I mean, Botox is a classic example. But those are ones where they consider them to be more line extensions, and it's managing a franchise, and it's taken into consideration from the perspective of how can I maximize sort of my return on investment. There are companies that are out there, specialty pharma companies. Unfortunately, some of them had a bad rep recently. But they are actually taking the repurposing approach and coupling existing drugs with new indications, but then also formulating them so that they're delivered in the right fashion, at the right dose, in the right place to the patient. And that sort of changes the nature of the drug significantly while still leveraging the existing safety of the drug. And so that's, our view is that's actually a great way to expedite getting drugs to patients, because you are going to run out of, the time to discover and develop drugs when you've got so many patients with so many variations, and it's just not a feasible problem. And so leverage what you've got.
Designing clinical trials is very hard.
It is. I mean, this is an area, like from a drug development perspective. How do you do clinical trials in the era of precision medicine, especially in pediatric oncology? You may have a handful of patients who have a profile that best matches your drug. How do you recruit them? I mean, this is, again, I think, where big data approaches can help. If you can connect a Facebook or a Patients Like Me sort of consortia that can tap into all of these patients and enroll them in the clinical trial, that whole process can be speeded up as well. So
Talk, if you could, just very briefly, because then we'll need to wrap up and give the audience a chance to ask questions. Drug design. Let's say you know a sequence. How do you design a drug intelligently, knowing that that's what it needs to accomplish?
Still a hard problem.
It's still a very, very tough problem. That's sort of the holy grail in the pharmaceutical industry. We may be able to get efficacy, but then we may run into unexpected safety problems. Or sometimes you'll have a very safe drug that just doesn't work. It is a problem. I mean, people are laughing, but the average cost of getting a drug to phase 2 is $50 million. So you're taking a $50 million bet that your drug is actually going to work. And so it is a hard problem in the industry.
But if it's designed, it should work. If it's
there are companies that are using computational chemistry approaches, so applying big data from a structure perspective.
Exactly.
And ideally, you'd want to marry all of these different endeavors to end up with the successful drug. So I think that effort is going on as well.
So we should mention there's a new kid on the block. There are drugs now that work not in the traditional way of attacking a specific mutation in the tumor, but these so-called immunotherapies, which were mentioned in the moonshot speeches and have been really gaining a lot of headlines, or specifically designed to activate the immune system so that the cancer can't hide from it anymore. And that is, this is a more generic approach. So it isn't one mutation, one drug anymore. And so I think there are other ways of thinking about this, and in particular, the immunotherapies have an enormous potential and may be part of this inflection point that you hear now out of Washington, and of course, Francis Collins has specifically predicted that we may be at an inflection point in cancer. So I always want to remind the audience that there are other avenues, and it isn't a hopeless, billion-dollar per drug slog through thousands and thousands of cases. We could have transformational ideas here.
Thank you. So this is time for anyone who has a question to go on
You
up
have
to
a
the
bunch
mic,
already
if you
here.
could, over on the right-hand side. There's someone point out the microphone, thank you. And while you're walking up there, let me ask if there are applications outside cancer. This evening we've been very cancer focused. Those of you, I realize.
Oh, absolutely.
Anyone
Yeah.
up here?
Atul is a
Yeah, so I
pediatric
think we have,
physician.
There's precision medicine is really being applied, big data is applied to a lot of different
Absolutely,
diseases.
yeah.
Infectious diseases, obviously we're all thinking about Zika viruses now, so
Yeah.
we're probably going to see a lot of big data around Zika in the next year, just a prediction there, but also diabetes. In some ways, cancer, believe it or not, as terrible as actually the field can still seem, it's ahead of other fields. Where we don't even realize there's subtypes of diabetes. We probably think of everyone as, you got diabetes, you treated kind of the same way.
Type 1 or type 2.
I know,
That's
maybe
it.
just kids and adults here. Boy, there's a long line of questioners here. Yeah.
First question, with the DNA sequencing, are methylation patterns being collected as well? And their correlation with disease? And if they're not being collected, are there any plans to collect?
Boy, we have an aficionado here.
They are being collected in research projects. They are not currently being collected in immediately contemplated routine medical use. But as we understand more of their role from a research, then we expect them to be part of clinical evaluation someday.
I mean, the sooner they can start being collected,
Absolutely.
even if they're not understood, the better.
So for a little background for those, so the DNA, we always think about A, C, T, and G, but the DNA can also be modified, in particular the C, cytosine residue, can be modified chemically, and that can have a different behavior then, it can have an effect, and so forth. So these are things that are called epigenetics, because they're, in addition to the basic A, C's, T's, and G's, there are other issues that one takes into account, and those are increasingly important. Good point to bring those up.
And actually, they are very important, pediatric cancers in particular. And so while we're not collecting methylation data yet in the clinical setting, we are collecting gene expression data, which could be thought of as a readout, like a consequence of some of the methylation changes. We know that it's not ideal, but at least we're collecting that.
So you're collecting
RNA
yes.
sequencing.
RNA and methylation.
Yeah.
So there was a passing reference to the recent news about computers playing Go.
Yes.
And then the discussion of drug design. And I wondered if any of you can comment on, is there any work going on to apply deep thinking, deep learning, neural networks to drug design?
Yeah, I can start. I mean, really almost all of us, what we're talking about is machine learning of different kinds here.
Right.
And for the aficionados, supervised and unsupervised machine learning, we typically use both of those kinds of approaches. Neural networks could be approached, there could be SVMs, there could be a lot of different approaches. Deeper layer machine learning, or deeper layer neural networks are obviously the rage right now. There are a lot of open source tools that have been released, so obviously a lot of different labs are using those. So there's no reason to rule those out. I mean, but there are also other ways to do machine learning too, I think.
Again, kudos to Google for a really hot machine learning package
Yes.
that you released.
Right, recently.
Yeah, I saw
released.
Microsoft copy and
Oh,
it's
yeah.
released theirs too.
Yeah, right.
Yeah.
I know, someone could still like Microsoft. Okay.
They have some great tools.
Yeah.
Really great tools too.
Yeah, thank you. Not being a computational biologist or a computer scientist, I need some translation about how you bridge a lot of what you talked about. Atul, you started talking about data connectivity and data interoperability and data exchangeability and the problem of actually getting information back and forth between parties to do this computational work. And then David, I remember you mentioned the internetting of all of these data using HTTP or TCP/IP or these kinds of things. And then of course we had the discussion about the cloud and big data and I guess IoT to some extent in this whole mix. My question is, why isn't this being bridged and what's stopping anyone from actually doing this work to make sure that there's greater efficiencies and effectiveness in collaborating?
Maybe I'll start. I mean, So we make this sound, you have a lot of kind of engineering types and scientist types here, but a lot of this is social engineering, right?
Yes.
So we have ways to solve this in a lot of different fields, and this field obviously needs to get solved as well. But 99% is getting the right people to want to solve it and then actually just come up with ways to do it. I think from the technical side, I think you're taking people that used to compete with each other, whether scientists used to compete with each other, now we have bigger projects where we got to work together. Health systems that used to compete with each other, maybe still compete with each other, have to share data. So I don't know how you guys would answer, but 99% is the social side of it, to get everyone to want to work together for common goals, right? I mean,
That's right. Absolutely, we have a silo mentality that we have to break with social engineering. So too many companies and institutions and other entities feel that their genomic data is somehow something they want to monetize. It's, whoa, it must be valuable. Let's keep it to ourselves and monetize it in some way. And that attitude is holding back. Maybe we'll make great discoveries. Maybe we'll be able to patent those great discoveries. Maybe, there are all kinds of reasons one might view the data as a precious resource that we shouldn't share, or at least we shouldn't give away, right? That's how much money can we get for it? Yeah. And we got to get beyond that because the key, we all hold a bit of the key, right? And it's only when we put it all together that key works. And so it's much more valuable combined than it is isolated in all of these silos.
I mean, in some ways, probably the pharmaceutical companies still have a long way to go to getting them to share data, right? How easy can you get to pharmaceutical data right now?
Absolutely.
They're definitely major offenders.
That's been a major challenge, and I'd like to point out I'm not representing the pharma industry. I'm just representing NuMedii.
Don't throw eggs here.
Yeah, but it is true. I mean, there's lots of information, there's lots of clinical trials that they've conducted. It'd be great to get our hands on that data, learn from that. I think there's lots of information in those clinical trials that can benefit new drugs that are being developed, so certainly encourage that. I would also add that I think part of the challenge might also be that we haven't had people who are cross-trained in different disciplines. So with Matt, a classic example of someone who's a computational biologist, really understanding these two worlds. So to stitch them effectively, they have one foot in each world, and being able to do that and having more people trained like that is going to be critical for us to make this process a whole lot easier going forward.
Yoda has a lot of that data. Yale online data.
Yoda. Yeah. They have some of that data. We know Yoda really well. But you guys are training undergrads like this too at Santa Cruz, right? That's the whole kind of thing.
Absolutely. Right? We had one of the first Bachelor of Science in Bioinformatics in the country. And we've been cross-training students so that they get both wet lab experience and computer experience by the time they're finished, and they understand how to talk both languages, which is really important.
I have two questions. One is whether in your data points, do you collect data from patients who you've already treated, and do you follow up with them and include that in your trials, especially for the Global Alliance? And the other one, it's very much related to the DNA methylation question. I work at NASA Ames, and we have, just here down the street, and we have a new project initiative called Gene Lab. And we call it Gene Lab, but we actually look at DNA methylation, we look at proteomics, metabolomics, RNA-seq, and these are from experiments, animal experiments or plant experiments or any type of experiment that's happening on the International Space Station. And the microgravity environment is known to affect the immune system, to promote stemness, there's leukemia due to radiation. So it's both a prompt, and I'm curious what the interest of this community is to look actually at that kind of environment and try and see whether there's any it's actually an exploratory approach, right? You want to try and look and see if that kind of environment that's completely unique. So it's you were mentioning stepping up. Well, that's really stepping up and looking at the that doesn't exist anywhere else. Right. So I'm curious about the interest in that.
Let me just address the first question because it's a long story about space effects. But the first question of how do we keep track of patients as they go through their treatment process and ultimately as they go through their lives. And this has been a little bit of a privacy stumbling block, but it's a good example of how technology can help. So normally you would think, well, we want to be sharing these data, and so if we want to have the data out there and follow the patients, then we would somehow want to be able to recontact the patients, and then that would mean that there would have to be personal data within the database, and then that creates liability, and it's a privacy threat, and so forth. But it turns out that we have designs now for databases where people can anonymously contribute to the database, and then they can update that data, and they do that by getting basically a digital signature that identifies them secretly as the owner of the data without revealing it to anybody else. And it can be proof of ownership of the data, so you can't have somebody else coming in and corrupting your data. So you can control your data in this shared resource and still remain anonymously. These are just a few, still remain anonymous. It's just a few nice little tricks cryptography brings to this, where we can treat some of these privacy issues and still have lifelong contributions to these data.
Space? You're gonna.
I'm gonna boot on the space question.
These privacy issues really come up for us. There's a twin study going on right now, and hope it's at Stanford if the data isn't posted at Stanford. So the data hopefully will be posted in GeneLab at some point. So we're creating an open repository as well of tools and bioinformatics in the cloud. So of course the question there comes about privacy, but there's very little privacy out there because there are very few people, so they're very identifiable. So it doesn't it's not going to work. So that's the kind of things we're running into. So when you talk about rare diseases, that's also a problem because somebody who has a rare disease may be easier to.
But so one thing I've noticed from the parents and families with people with rare diseases, not to generalize, but they're usually the first to share everything with the world.
Yeah.
And anyone who can help gets a look at anything, right? At least the family members that I know.
And
that's right.
The astronaut corps as well. People, they just want to share.
Yeah.
Right.
Anyone that can help. So.
I want to second that. I'm George Church. You can take my phenotype and my genome.
He's not really George Church.
I wanted to.
Not everybody got your joke, Mark.
No, yeah.
There have been many breakthroughs despite some of the challenges in cancer genomics. So in melanoma, the BRAF inhibitors and immunotherapy and so forth. And I wanted then to bring up neurological disorders. So for ALS, for example, since Lou Gehrig, there's been one drug that prolongs life about 2 months. And so my question for you all is, based on your experience in cancer genomics, how would you reboot and start again in a disorder like ALS with your experience and advise some of these organizations who are approaching this with, say, 300 patients' genomes? And I hear that and I just sort of weep because I think that all the best intents and so forth, that there needs to be additional focus on that. So your advice on how to approach this?
Well, okay, so I'm going to say one of the major things we want to do with the Global Alliance is to engage patient advocacy groups and any group that is involved in a particular disease because they're passionate about that disease. Give them the tools to share the data, not have them all invent their own tools for analysis and so forth, but give them the tools they need so they can take their data in place where it is and convert it into an API that will allow sharing and interactions. And we're doing this with different countries, different institutions, and so forth, and different disease areas. And these groups are very energized to want to increase their numbers. And when you become part of a global network of data sharing, then that's how you find other people in the world that are similar to you. One of the Global Alliance sponsored projects is called Matchmaker, and it simply is a genetic matchmaker, also a phenotypic matchmaker. And so doctors can get on the Matchmaker network and say, boy, I got a patient today, and they have these really unusual symptoms, and we looked at their genome, and they're missing a megabase of DNA on chromosome 17 in this spot. And I don't want to have to do what I always used to have to do when the mom comes and says, well, you just told me about my 2-year-old. What is it? And I have to say, well, I've never seen anything like this before. No one like this has ever been treated in our hospital before. And that's the end of it, right? It doesn't have to be the end of it. You should be able to get on this international network because maybe there's a kid in Tokyo or there's a kid in London that had the same condition, and that will allow you to do that kind of matching. And this cuts across all of the different disease areas. So we want to mobilize all of these patient advocacy groups to get on these networks that we're creating through the Global Alliance and get in touch and amplify their research and power.
And another exciting thing, just to add to the importance of advocacy, is that there is this new movement that will allow a patient to actually own their data. So if a patient goes and gets genetic testing, they can actually go to that genetic testing provider and request their raw data, and then they can share it with anybody. They can share it with the Global Alliance or another hospital or what have you. And so we as scientists, I think, have to educate people better, educate patients about what they can do and the importance of getting access to that data and what can be done. And this is where advocacy groups would play a key role as well.
It was a good start of the meeting. When we saw the first video, it was very heartfelt. And you can associate only when personally something hits you. And that's what I found myself. And to the extent, as Dr. Butte said, there's never late. And here I'm standing as an example. I am trained as a physicist, scientist, and did system design analysis for my ex-employer. The reason I said ex-employer because for the last 5 years, I have been studying genetic code, gene expression, trying to find out a cure for Mendelian inherited disease, which my daughter suffers. However, it's not life-threatening. It's a question of managing it. That's what is going on. When I look at OM-IM site, everything is well documented. You know, you go up to the amino acid level, 8. I have not seen a single Mendelian genetic disease cure. I'm wondering what's the problem? We understand very well, it's well documented. Why are we unable to do that? That's my first question. Second one is, going back to coding level, that's what I'm looking at right now. We know which amino acid is wrong. In order to correct, you have to use a proper codon. Codons are degenerate. My question to the audience here is, out of those degenerate codons, which one should I use to correct that amino acid?
Yeah, so you're looking for us to cure a disease in about 2 minutes here. So let me try this, right? Look, so Mendelian diseases, just to explain to the rest of the audience, are these kind of disorders where, you know, single misspelling or alternative spelling yields a constellation of symptoms that's pretty classical, meaning that everyone that we kind of see has that spelling difference gets this disease. So much so that they run in families, and those were the easiest to kind of find those genes. So for example, sickle cell disease, cystic fibrosis are some of these examples. Those are kind of easy examples, but they're also hard ones. Cystic fibrosis, there's one gene, one recipe, but it's got 200 ways you can spell it differently, and all of them cause slight differences in what we call cystic fibrosis. You know, but I'm picking cystic fibrosis because at least there's some glimmer of a hope of a drug for that one. Okay? What I mean is this era of precision medicine where we do have at least one drug that helps treat 4% of cystic fibrosis patients. So you're taking a rare disease, 4% of them, but there is a drug that works for them. Now, it's not perfect. It's super expensive. We kind of justify that because it took forever and a lot of money to get that one. And maybe it'll be useful for others. But in some ways, that's your one piece of hope here. But it took a long time to get to that one. We've got to be more efficient about it. We've got to make use of the data and the people and the samples and all this. But we also, you know, again, we talk about human studies, but it also helps to have other models to work faster. Some people don't like animal testing, but mouse models that resemble the human disease, If you can kind of create that, you can move a lot faster. Another piece of hope are these new types of drugs that can snip out different parts of DNA and replace them, something called CRISPR-Cas. Despite what you read, that's a UC Berkeley invention.
Yeah.
Despite what they say, that's a UC invention there. But so there's some hope there, at least. But you got at least one, okay? You got 4% on that one. So, and that's just 2 years ago. So long way to go. I think there's 3,000 Mendelian diseases. I just heard the number about a couple hours ago. So now there's 2,999.6 of them left, right? So
Yeah, I just want to add that, as you say, cystic fibrosis, the one I'm looking for is the same transport gene involved, which I'm concerned with.
About 2 more questions, I think
that's a lot, huh?
Yeah, I have a question that might be like totally off the wall, but I was thinking.
We all are here.
The whole gene therapy thing, I mean, you're talking about sequencing genomes. Is there a gene therapy that might, be helpful here?
Yeah, so what gene therapy means a lot of different things to a lot of different people. I think Alejandro can certainly tell you we can routinely take cells out of people, change a little bit about them, and put them back to help fight cancers, to fight different diseases. I saw a pediatric immunologist here somewhere at one point. We can replace immune systems, and so David Lewis was here earlier. So I think that kind of cellular therapy, gene therapy is routinely done. But to replace your entire DNA or just to target certain tissues is still beyond our technology right now in general.
Absolutely.
Well, I think the thing to remember about cancer is that cancer is almost never a disease of one gene.
Yeah.
Right? So it'll be very difficult to conceive of how you would use gene therapy to treat cancer, unless you could, maybe modify the patient's own cells to attack the cancer. That's one possible way.
You can think of some of the more sexier, provocative immunotherapies is thinking about some kind of gene therapy where we may be able to modify the immune system to attack the cancer. So it's only, but as Atul said, we're modifying only certain cells, the cells in the blood system that are derived, the immune cells that are derived from this hematopoietic lineage. So you can, that's easily changed, but to change a whole liver or to change a whole brain, all the DNA in that's not feasible in any foreseeable
in some ways,
future.
that's kind of why we like to look for molecules, drugs, chemicals that can mimic what that would do in effect, because doing the change is actually so hard, right?
Yeah.
Last question?
In the next decade, where can we expect to see the most additional progress in understanding the genome? Coding variants or non-coding variants?
Non-coding variants.
I think that was
Coding and non-coding. We've got to explain that difference first. All right, so here's the difference here, right? So, I mean, David has, these guys have their analogies for what is the genome. So let's end with what is the genome, right? So to me, I use the recipe book called The Joy of Cooking, right? How many of you have The Joy of Cooking, right? It's the definition of wishful thinking. We think you put it in the kitchen, it's going to cook itself. We have this, doesn't do anything, right? But everyone, I don't use Bibles and things like Korans, but Joy of Cooking everyone relates to, right? So The Joy of Cooking has a certain number of letters, and the genome, we're talking about 3 billion or 6 billion, depending if you count your mom and dad together. It's about 2,000 copies of The Joy of Cooking. So Joy of Cooking is pretty wide, and The Joy of Cooking, 2,000 of them is pretty tall, 2.5 statues of liberties tall. And so that's about 6 billion letters. And same thing with the genome is there's recipes, right? So we call them genes, but these are recipes. And so the question is, you got the recipes in The Joy of Cooking, but actually there's a lot of figures and how to set your table right. If you actually look at the book, it's kind of like Martha Stewart-like, like put the fork this way, a spoon. But those aren't the recipes, right? So is there more meat in the recipes or around the recipes? The recipes are actually only 1.5% of the genome. The rest, 98.5, is the other stuff. So I just heard another good geneticist, Rick Lifton, speak at UCSF. I just drove down here. And he's a pretty good scientist, and he told me the future is going to be in the coding variants here, because that's where they found most of the past ones. Now that's a kind of circular logic, because they look there, right? I mean, the recipes, you can tell if a restaurant's good or not based on the recipes, right? Yeah. So if you keep looking at recipes, you see problems with recipes, you think all the problems can only be in the recipes. So it's kind of circular, but they've had a lot of luck looking at the coding regions of the recipes. I don't know if you care to argue. I have no other argument here besides talking about cooking. But
Well, you know, as you said, 98.5% of the genome is non-coding. And we know that all of the regulatory logic is in there, how to turn genes on and off. And it's the orchestration of the process that will be very, very critical. So when we're discovering disease-associated effects, now that we start to look at that non-coding region, it is opening up new worlds. So I think you will find the so-called dark matter or junk DNA, all these pejorative terms that have been administered to this part of the genome, actually, you know, that will be increasingly important in our medicine.
Last question.
Oh, no more.
Mike gets the last.
All right.
I'll bring up the last question.
Okay.
So I'm just kind of wondering what those of us who don't study this field, what we can do, and it's a kind of a question for Olena. You mentioned the Treehouse project. I'm wondering if you can tell us a little bit more about that and how we can help.
So the Treehouse project is an international project that, so CKC that we mentioned, California Kids Cancer Comparison, so that's a California-specific project. We're partnering with California Trials, and we're hoping to help 150 children in California. And so Treehouse goes beyond California, and that's where we have partnered with hospitals around the country and also in Canada. And we're hoping to offer this approach or enable this approach where we compare every child's genome against all the other genomes that we have, and we will have through Global Alliance, including adult and pediatric genomes, to see if we can learn from these comparisons.
Can I just put in a last word? How can you help, right? Let's just at least address that point. Help the researchers around you, help the campuses that are represented here, help the hospitals, help your representatives in the state know that precision medicine is a good thing, help your representatives in the country know precision medicine is a good thing, right? Every one of us can have some step there. And I think that's the important takeaway, is you can actually make a big difference today. Instead of thinking of this as budget dust, make it bigger, right? Because they need to hear from you.
That's right.
Great take-home message. Thank you.
Are accepted at treehouse.ucsc.edu.
There you go. That's right.
All right.
Many thanks to you in the audience for just elevating and expanding this conversation. It's just been fabulous. And thanks again to our panelists.